The overall goal of this technique is to utilize endothelial cells as a novel type of planar antigen-presenting cell model to investigate fundamental antigenic signaling processes.
This method can help us address key questions in the field of adaptive immunity, such as how subcellular and molecular remodeling dynamics are coordinated spatially and temporally within the immunological synapse. The main advantage of this technique is that the endothelium forms a virtually planar APC surface. This allows for imaging with optimal spatiotemporal resolution within the context of a physiologically complex and deformable cell surface.
Though this method focuses on CD4 positive Th1 effector memory cell activation, it can also be applied for the study of a variety of T cell types, antigen, and signaling processes.
Today, Chris and I will show you how to perform this technique.
Begin by co-transfecting primary human lung or dermal microvascular endothelial cells with membrane YFP and soluble DS-RED via nucleofection. And plate the transfected cells onto live cell imaging culture plates. The next day, replace the cell culture medium with fresh medium supplemented with interferon gamma to induce MHC2 expression on the endothelial cells, followed by stimulation of the cells with 20 nanograms per milliliter of TNF-alpha 24 hours later. On day 3, 30 to 60 minutes before starting the experiment, incubate the endothelium with 1 microgram per milliliter each of the bacterial superantigens Staphylococcal enterotoxin B and toxic shock syndrome toxin 1